Okay, so I'm going to start making less and less announcements in class. They'll be posted, right, so that anybody has access to them at any time. So under our modules, right, our first test 1, module 1, right, is all this information. It's a lot of stuff. There's a new discussion board. I already saw some of you guys have posted. That's great. Hopefully you followed the instructions so you don't lose points. Remember, all discussion boards you post and then you reply to 2 classmates. We didn't finish up microscopy. I thought we did, but looking over it, we didn't. So we have 1 objective left. We'll go over. I wanted to point out these animations and videos under the first module. So those of you guys that are visual, right, type learners, these are great tools. This is the gram stain animation I showed. This is me staining in lab, right? Not required, but you know, if you ever have to take a lab class, there's some good tips in there. Make sure you can do that procedure. Prokaryotic shapes. This is a video link, right? You'll notice it opens up on another page. This is actually from the publisher now. They've got links for me instead of having to use the YouTubes for some of them. And this one relates to the last thing that we're going to talk about today, which is why I was like, I think we didn't get to something yet. So this is a good one to watch. I'm not going to play any of these. Again, these are all links to the publisher's provided website. And these are all embedded in my PowerPoint today, but I'm not going to play any of them. So there, you can either go here, or if you download my PowerPoint and you show it in presentation mode, the buttons actually work. But if you just want a quick reference, they're all here. And I had posted an announcement, and hopefully that you watch these before class today, so you'd be familiar with some of these mutations ahead of time. Actually, take this thing out so I'm not lisping. Here we go. Okay. All right. So those of you guys that come in late, to make sure you check with me after class. I mark you off on the attendance. Okay, so going over to today's PowerPoint, if I click on the right thing. As I said, we need to finish up microscopy and cell structure. The last objective that we hadn't got to is identify the process of endocytosis and exocytosis in eukaryotes, and you can see I refer you to the animation too of lysosomes. So it goes through step by step the process that's outlined in this diagram. I don't know why my slideshow thing is not showing up. It's one of those days. Oh, I know. Oh, from the current slide. There we go. Hallelujah. Thank you. The usual button wasn't showing up. Okay, so only eukaryotes have this ability, right? Prokaryotes like bacteria and archaea do not have this ability. One of the main reasons is they have a barrier that's in the way, and that's the cell wall, right? All prokaryotes, for the most part, have a cell wall, except for a few exceptions. So you can't engulf something from the outside using your membrane if you have a cell wall in the way. So only eukaryotes, and especially ones that, of course, don't have a cell wall, because there are some eukaryotes, like plants, algae, and such, have cell walls, don't have this ability. We have specialized white blood cells that we're going to definitely focus a lot on in our next unit that can do this process of engulfing. And for them, it has a special name. It's called phagocytosis. And the most part, we're engulfing bacteria, foreign substances, viruses. And the same thing, just like an amoeba moves, right, and that's how they engulf their food too, is they're going to use their cytoplasm and push it around and form these what's called pseudopods, or false feet. It's also how they move. So they're going to surround and engulf stuff. If it's large stuff, it's phagocytosis. If it's relatively small, it's referred to as penocytosis. These are all forms of endocytosis. Endo meaning to take within the cell. Depending on what the cell engulfs, it may need to digest it. And so it'll do that in specialized membrane-bound vesicles, because remember, only eukaryotes have internal membrane organelles, like the nucleus. Lysosomes is an example of that. These contain digestive enzymes. You don't want these running free inside the cell. So they're encased within a membrane, right? This specialized vesicle. Notice they give a special name to the ones that are taken in by penocytosis. They're referred to as an endosome, right? Some referring to these vesicles. Phagosome, right, by phagocytosis. A lot of times these endosomes and phagosomes are going to fuse with a lysosome that contains the digestive enzymes, and they're going to break down whatever's within that vesicle. So digestion occurs. And what they do with the name is they just fuse it for phagocytosis, right? It becomes phagolysosome. It's a phagosome fused with a lysosome. So guess what they do with the name? They fuse it together, right? And then sometimes the cells will spit the stuff back out, right? Exocytosis. Exo meaning leaving the cell. And so these are large waste products and stuff that can't easily go through the cell membrane. They actually encase it into a vesicle. So how are these vesicles, especially a lysosome, made in the cell? That animation goes over it. So if you remember some of the organelles, important organelles about eukaryotes, and again, prokaryotes, no organelles, right? They have DNA, it's in a region, but it's not encased in membranes, right? They have other structures within, but they're not membrane encased, okay? And of course, they've got tons of ribosomes. All cells have to have ribosomes, because what do ribosomes do? They make proteins. But what do we learn about prokaryotic ribosomes in comparison to eukaryotes? They're smaller. What are ribosomes made out of? What molecules are they made out of? How'd we end up with the three domains, bacteria, archaea, and eukarya? Why aren't bacteria and archaea linked together? They're both prokaryotes. Their ribosomal RNA, remember, is different, right? That sequence within these ribosomes is different for prokaryotes. It's different for bacteria versus archaea as well as eukarya. The protein component of the ribosome is different for the different groups of organisms that we know. And then much more complexity when you look at eukaryotes, right? So these are two examples of prokaryotic cells. So a little bit review from biology class, right? Ribosomes are not organelles, but other organelles are like the mitochondria, right, which are necessary for energy. Electron transport chain is located there, right? Glycolysis happens outside, and then electron transport chain. You have the endoplasmic reticulum. There are two types. Anyone know why the rough ER is called the rough endoplasmic reticulum? It has ribosomes attached to it, right? So that one's really important for protein synthesis. Things that are processed by the cell are done so by another set of stacked membranes referred to as the Golgi apparatus. So a lot of times the proteins and stuff that are made in the rough endoplasmic reticulum will travel to the Golgi apparatus to be packaged together to form, say, a lysosome. And one cell I wish they had here, which would be another example of a eukaryote that's microscopic, would be an algal cell. And what organelle does that cell have that allows it to be able to produce energy from light? What organelle uses light energy, does photosynthesis. Chlorophyll is the pigment that traps the light. Chloroplast is the name of the organelle, right? So that's another important one. And I'm actually gonna, because I want to keep these separate, I'm going to